stick to schedule. We're going to try to keep this talk to schedule. You might notice that there's some buffer leeway times in the program. Please don't try to use those times for your talks. Just extended coffee break, extended lunch, but please don't try to use those times. Okay. So anyway, our first speaker is Eduardo Sontag, who will give us a very interesting talk about a mathematical model of evolution of drug-induced resistance.
Okay, thank you. So I got 100 slides, so I'll have to go a little bit fast, but that's okay. Anyway, I'm going to give the same talk basically in 2 weeks here, but an hour talk. So, you know, it can stay for 2 weeks. Before I forget, this is joint work mostly with Jenna Geberts, whom you all know and is here, is talking, I think, tomorrow, and Jim Green, who was a student of Doran's, okay, and there's a couple of other people involved, but they're the main people in the project. So what got us into this this project was this idea that we started working on. Oh my God, was it 2016? Whatever. It's like even earlier. Okay, so about 10 years ago. Jenna came as a postdoc, as a sabbatical visitor at Rutgers, where I was at the time, and we started talking about this interesting question that most people think of resistance to therapy, whether targeted or chemotherapy or whatever. Most people think of resistance as classified into pre-existing, sometimes called standing variation, or resistance that arises during treatment, sometimes called de novo resistance. And if we go back to Luria Delbruck in bacteria and so on, that's sort of the classical thing that everybody studies. And what we were focusing on was the fact that, in fact, we need a finer distinction because, as many people had pointed out around that time, there's also some sort of Lamarckian as opposed to Darwinian process that occurs where the actual drug that you're applying changes the generous variation, if you like. So in other words, instead of Darwinian selection, we have a process in which we both generate variation and then do Darwinian selection. And the question is, okay, so what? You talk to biologists, they say, well, who cares? I mean, so, you know, in the end, it's the same thing. So what we're trying to do is to try to say, well, mathematically, we can actually differentiate between the two in a way that I'm going to tell you in a minute. And then we could ask, does that really mean anything in terms of, for instance, how you would treat the disease? Okay, it's a cancer. Okay, so I'm going to skip obviously a lot of stuff because I can't. But the paper that got us started with all this was this paper from Shui Huang's group. Pisco was the first author, and in this paper what they had is they showed that for a particular leukemia cell line and for a particular chemo agent, although there were some other examples too, but they showed that resistance is reversible. Okay, even at the single cell level, you can track and see resistance is reversible. These experiments I don't have time to go through in this short version. And reversible in the sense that when you withdraw the drug, the population re-equilibrates to some mixture of resistance and sensitive cells. So somehow, the fact that you're applying a drug causes a shift in your distribution in cells. And when you take out the drug, the distribution sort of goes back to the original sort of wild type distribution. So how do you model this? What does it have? What does it tell us about the about the system? How can we do something useful with it? So the idea is very, very simple. We take the simplest possible model. In fact, I'll have an even simpler model as I go on. We take the simplest possible model. We have logistic growth for a sensitive population, logistic growth for a resistant population. As yesterday in one of the lectures, we assume that the rate of growth of the resistant population is slower than the rate of growth of the sensitive population because the resistant population is typically expressing some mechanism of resistance. For example, in the case of MDR1, pumps that will be taking drug, which is what we studied there, pumps that are sort of pushing out of the cell the chemo agent. So, and that of course has a fitness cost. So we assume that RR is less than R, and we also assume that DR, the rate of killing of drug-induced killing, is less for resistant cells than for sensitive cells. So we have this setup, and what I want to draw your attention to is I'm going to let gamma be equal to 0 soon, just for simplicity in the mathematical analysis, but there's some back transformation. But the main point I want to draw your attention to, either killing terms, right, from the drug, these are killing terms from the drug, but I want to draw your attention to epsilon and alpha. So if you forget about alpha, we have a negative epsilon s, a plus epsilon s. This is a flux from sensitive cells to resistant cells. There's a gamma there, which is the backward flux. So this is occurring independent of the drug. So think of it as random phenotypical change, which is known to happen. In fact, because of those steady state probability distributions, you know that it happens, and the experiment that shows that it's there. But in addition to that, and it could be phenotypic change, of course, could also be mutations, right? But the thing, I mean, epigenetic or genetic leading to different phenotypes. But I also want to draw your attention to this alpha term, which is a dose-dependent switch, right? We have a flux, alpha US, mass action kinetics, just for simplicity, alpha US into, right, negative flowing into the second compartment. And this flux, think of a valve, right? This flux from sensitive cells to resistant cells is being controlled by the drug. So that is a term that we think of as Lamarckian resistance. In other words, a term that is controlled by the drug that tells us how sensitive cells are being formed. Well, the epsilon term is the one that corresponds to things that are happening sort of at random, if you like, okay? So that is really the whole, the model. And then we ask, okay, fine, anybody can write a model, but then, what's the point? I mean, that's a trivial exercise, writing models. The question is, what do you do with it, right? So what we did is we said, well, let's set up let's have one control problem. So in a simple control problem where maybe gamma is 0, just for simplicity, I'm also assume complete resistance for the resistance as the R is equal to 0. In this model, you can see it's always going to happen that everybody's going to end up, in the, it doesn't matter what you're going to do. This is, I'm normalizing to 1, so the maximum positive S plus R is 1, and S and R is a proportion of resistance to sensitive cells. You're going to end up basically with everybody becoming resistant, and that's going to be the end of the story. So, but what you can ask is the following question. You can ask a question, and this comes back to something we were talking yesterday in one of the groups. You can ask the following question. Let's try to make the time to progression as long as possible. So the particular game that we're going to play, we're going to ask the following optimal control question. We're going to say, we're going to start treatment when there's a detectable level V sub D of the total tumor, S plus R, and we're going to stop treatment when we get a critical point, resist criteria, whatever you want. That's just purely mathematical. At some point, we're going to start treatment. We're going to increase the time up to here as much as possible. What is the optimal strategy for that? And how does it depend on whether we have Lamarckian or purely Darwinian mechanism? In other words, does it depend on alpha being equal to 0, which would be the purely Darwinian thing? We're just selecting on the basis of the difference between the DR and the D, or does it depend on alpha, which is this induction rate, this Lamarckian effect that we like? So I obviously don't have time to go through the details. There's a huge amount of optimal control theory, differential geometry, Lie algebras, all that good stuff that happens when you do optimal control theory to prove theorems about these things. Okay, you can read the papers for all that. But I'll just give you sort of the sort of some of the bottom line idea, intuitive ideas. So the basic thing is, we ask the following question. Suppose that we compare a continuous therapy where we apply the drug continuously in the time between detection and the time we give up, or suppose we apply some sort of pulsed protocol. Okay, let's compare those two scenarios. Okay. The main question is, if we compare those two scenarios, is there a difference between the Lamarckian case, the case where we're inducing resistance, we're inducing variation in our ecological system, okay, as opposed to simply doing selection. So if we ask that question, at least for this toy model, we get the following bottom line conclusions, and I won't go through the details, but basically we get that if we have that alpha is equal to 0, in other words, if we don't have any induced resistance by the drug, if it's purely selective, okay, just purely Darwinian, in that case, a constant therapy is better. On the other hand, if we have induced effects, if alpha is different from 0, this is just numerical, with some values of alpha, one could do much more parameter studies. We did, but just to give you an example of what we expect, you actually get that pulse therapy is better. So bottom line from this part of what I told you is you could play this game. A biologist, when I talk to oncologists, they throw up their hands and says, who cares if it's induced by the drug or not? What matters, what happens to the patient. But the point I want to make is, yes, that is true. But from the point of view of differentiating between the induced and the purely random or whatever spontaneous changes, the whether one is happening or the other should have an influence on therapy. Now, this is not a realistic model, but it should have, it will actually make a difference in kind of therapy. So it's sort of a meta principle, okay? I sort of like in my research this kind of meta principles as opposed to, studying a specific, 100-dimensional model. So as I said, we set this up as an optimal control problem. There's some examples of the optimal trajectories. Basically, for those of you that like optimal control, we're trying to solve a viability problem, which is not a standard optimal control. I haven't found in any textbooks in optimal control. You want to stay as long as possible in this set where you're under V critical, okay? That is not a standard, as far as I know, it's not a standard optimal control problem. We had to develop things from scratch. So it's actually cool optimal control theory, new math that comes out of all this, okay? And basically the trajectories end up being something sort of a bang control until you get to this thing, and then you have like a sliding mode for those in control theory, sort of a sliding mode control that allows you to stay on that, and then at the end you give it a little extra jump. So this is too much, just for a lecture, but I'm saying just, that's sort of the sliding mode trajectory that you have. And what we find is, roughly speaking, that the bigger that alpha is, the bigger that this induced treatment is, the more bang switches you need. So it sort of tells you something about the structure of the optimal control. We also have identifiability results. I'm going to skip that. We did a quick data fit to the of this model to the Pisco data. It actually looked quite good. I don't have time to go through the meaning of all these graphs, but just things sort of look graphically, to the eyeball metric, they look cool. We're fitting with this very simple model, which has very few parameters. We can fit this data. We cheated a little bit. There are a couple of data points that we couldn't fit and censored them out, but they were obviously wrong. I mean, there's some experimental error because they were like totally off. It didn't make any sense. So, I believe math more than I believe experimental data. Okay, sorry. If the math tells you something elegant and experimental data doesn't fit, there's something wrong with experimental data. Okay, I'm serious. I strongly believe in that. Okay, so anyway, so we had all this, and we had some really interesting. I won't talk about some other things, but let me just say that lately, and this is a paper that we now have a paper under review. We were motivated and started working with Natasha Comandante-Lu, who had been working with Falaia Shikhani and Peter Solger's group at Harvard in the past. And we were interested in understanding, is there a model that would fit some more interesting data, right? So we had a really good set of data and that we were using. And here's an example. I think I had the details of it's a melanoma cell line, again, treated with a different, with a targeted inhibitor. It's a BRAF inhibitor. Okay, so this is targeted therapy. The other one was classical chemo. And what we found in this case was that basically these are different doses that are being applied. This is experimental data. We found that basically there was no effect of the drug until about 20 hours after this is in vitro experiments, okay? Culture. There was basically no change. So we said we need to put some sort of delay in the system. I'm going to skip the actual the details of how we derived these equations. It's in the previous slide. It's important for other reasons when we get to optimal control. But basically, those are the equations that we have. We took the carrying capacity to be infinite. Okay, so it's just a linear system, no logistic term anymore. Basically, we have this. It's really, if you look at it, it's a time-varying linear differential equation. What could be simpler? Okay, time-varying linear differential equation. We fitted this gamma 1 and gamma 2 to 0.1, I think it is 0.01 based on biology. The rest of the parameters we let free. For each of the doses, we allowed the parameters to be different. And these are the fits, which for me are amazing. Okay, these are error bars over 4 experiments. And our fits fit with this very simple model, fit the data very well. Now, when we did this, we did it at 3 doses, then a log of negative 1.5, log of negative 0.5, etc. So I said them before. There were another 2 measurements that we didn't use in the training. When we use these parameters that we got from here, interpolated the values of the parameters for the data that we had not used, not censored, data that we had not used in training, we actually predicted, I'll get back to this in a second. We actually predicted pretty well the behavior for the data we hadn't used. But the most interesting thing about this is the parameters are very identifiable. First of all, there's a mathematical proof of identifiability using Lie derivatives, blah, 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 the usual thing. but for structural identifiability. But we also did a practical identifiability study where we picked the 5% error, the things that were within the 5% of the optimum with multi-star with a multi-star con, and ask for those word parameters give you a good fit, a reasonably good fit, and we get extremely tight distributions of parameters, okay? Extremely tight distributions, coefficients of variations like 0.05 or something, okay? So this gives us confidence that this is a good model for the data that we had. It's at very identifiable parameters. But then we said, well, but in the paper, they actually have a population of so-called quiescent cells. And I'm not going to go through the details. They do some single cell measurements. There's cells that stop dividing for a long time. But then we said, well, what happens if we put the quiescent cells in the mix? So we have this new model with quiescent cells because it seemed that that should be better. It turns out it's not better. We get the exact same fits with the exact same errors. And what's sort of interesting, and I know Jana loves this, the next slide, I did add it. You asked me to add the slide, and I did. We see that we get huge non-identifiability of parameters. So while before we had very tight parameter distributions, now we have parameters that could be all over the place and still give you the same result, which means that this model is not a very good model from a mathematical point of view. And I think that, yeah, optimal control therapy, I don't have time to go through that. These are the papers. The last one is under, yeah, there's some cool optimal control too. But those are the papers, and you can find them in my website. The last one is an archive. It's an archive, right? Did we put it in archive? We did. So the last one is in archive, and it's submitted, okay, to MPJ. So that's it. Wow, actually almost. Okay.